Okay, it's the secret life of water, E equals H2O, and I hope E equals H2O will become clear as I go on. So let me start with a common observation. You see a cloud up in the sky, a single unitary isolated cloud, and you ask the question, how does it come into being? Because the water, as you see from the slides, is evaporated from all over the ocean, or the lake, or what have you. And somehow the molecule, the aerosol droplets, or whatever comes up, and it must be directed toward the cloud. So how does this happen? I'll get back to that later on in the talk. So I want to talk about water, and the easiest way to think of a water molecule is as a dipole. It's got a plus and one and a minus on the other end. And you could maybe represent it, if you wish, as a single beam with positive and negative charges on the two end. Now, that's the question. I think, is that better? Okay, I hope if it sags, please let me know. Okay, so what happens then if one of these dipoles, yeah, it's better. Yeah, that's better. So what happens if one of these dipoles then reaches a surface that has charges on it? You might expect that it would absorb onto the surface, and if you have other dipoles, they would absorb as well and form some sort of layered structure. You have a lot of charges on the surface. However, the general thought is that this layering of water can extend no more than 2 or 3 molecular layers, because when you get much farther from the nucleating surface, you have the effects of thermal motion, which would disrupt any order. So the current view, if you read any textbook, will be that next to hydrophilic surfaces or charged surfaces, you have just 2 or 3 molecular layers of ordered water. What I'm going to present to you today is evidence to show that that's wrong. And that what really happens is that this ordering extends extremely far from the nucleating surface. And that this has huge implications for everything we think of, especially charges. And the first experimental approach to see how far this might extend is to set up a system, as you see on the slide, and then see how solutes distribute. So what you might expect is that in a region that's ordered, for energetic reasons, the solute should be excluded. And farther out, where the water is not ordered, you'd expect that the water is able to hydrate the solutes. And so you have plenty of solutes there because they're happy to be hydrated. So we set up a system to make this measurement, and the idea was that if we could find, for example, that there's a region from here to here that had no solutes, actually we use particles to start with. I'll get to solutes later. But if they were excluded from this region, then we might surmise that this region perhaps hypothetically could be ordered. So that gives an idea of the magnitude. What we did at first was to look at a gel. On the left side you see a polyvinyl alcohol gel. And the gel is sitting in a chamber. And to this chamber we add water and microspheres. The idea of these microspheres, little tiny latex spheres, 1 micrometer in diameter. And we wanted to see if there was a region in which these were excluded. And so the video looks like this, and you can see that what happens is that these microspheres march away from the interface, and they keep going and going and going. And finally they stop about 60 or so micrometers from the surface. Now 60 micrometers, in terms of the size of a water molecule, is practically infinity. So we were really shocked to see that maybe this region of ordering maybe could extend as much as 60 micrometers. But we thought maybe there's something special about this particular gel that gave rise to it. So we tried a different gel. We did the same experiment with a polyacrylic acid gel. We dumped the gel in the chamber, put the water and the microspheres in, waited 5 minutes, and here's what we saw. So here's the left side of the gel and the right side of the gel, and this is just an optical reflection, so please ignore it. So here are the microspheres. This is after 5 minutes, and marched from here to here, and left a gap on the order of 250 micrometers, 4 times larger than with the other gel. That's 1/4 of a millimeter, and you didn't even need a microscope to see it. You could see it with your naked eye. It was that large. So having witnessed this kind of exclusion, we had to give this a name, and we called it exclusion zone, or EZ for short. By now, this phenomenon has been repeated by many, many people with similar results. So there's not much doubt that the exclusion phenomenon is real. The question, of course, is what does it mean? So what I want to do, get into the base, is to answer 5 questions. The first is this really a general phenomenon or just some quirk of a few particular gels? Does it really arise, does this zone arise from is it because the water inside is ordered in some way or something else? Can water ordering explain the many anomalies that are well known to exist regarding water? And in order to create order, you need energy. So where on earth does the energy come from? Not obvious. At least it wasn't to us for quite a while. And might these findings apply more broadly? So that's what I want to go through. The first question is one of generality, and the questions you might ask is, what kinds of surfaces give rise to this exclusion zone? And then what solutes are excluded from it? So in terms of what surfaces give rise to it, the first thought that might arise for you is hydrophobic versus hydrophilic surfaces. Now, we tried hydrophobic surfaces and we saw nothing. Nothing that I showed you. We tried hydrophilic surfaces and we showed this kind of exclusion as a generality, as a class. I'll give you 4 examples of that. The first is hydrophilic gels. I've shown you 2 of them. Here are a few more, and there's still more that we and others have actually tried, and they all show these exclusion zones. The next class is biological specimens, and I started more or less as an engineer turned biologist. And this is a piece of muscle. And you can see on the left side that there's a region where there's not total exclusion, but very few microspheres and lots of microspheres far away. We've tried the inside of blood vessels, that is vascular endothelium. We tried plant roots. We tried cellulose from trees, and we see it consistently. So the second class, biological specimens, exclude. Then we tried monolayers, a single molecular layer. So on the left side, you see a single molecular layer, self-assembled monolayer. It's a standard kind of preparation on gold. And you can see that there is exclusion. And the reason for doing this is to see whether you really needed a thick sample to do it, like a gel, or a single molecular layer might be adequate. For example, if it acted as a template. The fourth category is polymers. We've tried many polymers. What you see here, this arrowhead shape, is a sheet of naphthalene, like a sheet of paper. Naphthalene is used often in fuel cells as the fuel cell membrane. So we take a sheet and we cut the sheet with a knife in this arrowhead form. We lay it down on the chamber, and then we pour in the microspheres in the water, which you can see here. And here's what happens. So the exclusion zone grows and it grows, and this is stopped prematurely, and it'll typically grow out to 300, 400, even 500 micrometers. We've even seen a millimeter with this on occasion. So in terms of generality, I've shown you that numerous hydrophilic surfaces show exclusion, but not the hydrophobic surfaces. Now, what's excluded? So I haven't shown you solutes, I've just shown you particles so far. And what I'm going to do is start with the particles and go down in size to solutes and small solutes. So in terms of particles, here's a list of some of the, you might say, particles that we looked at, although some might qualify as solutes. The distinction between the two is not exactly perfectly clear. Anyway, getting down in size, we tried protein, being in the biological realm, it seems interesting. We tried albumin. So we put the albumin next to some nucleating surface, and you can see here, this is a time series, and the top one shows a piece of naphthalene sitting in the chamber. The reason you can see it is because of fluorescence. And then in the second panel down, this is the time series. Second panel down, you put in the fluorophore-labeled albumin, and you can see the brightness. But you can also see, as a function of time, that the exclusion zone grows. So it looks as though the protein is excluded. We're talking about a few nanometers in size, perhaps 3 or 4 nanometers. Getting smaller, we started looking at dyes. The first one was sodium fluorescein, molecular weight 376. It was excluded. The next one we use are pH-sensitive dyes, and some of you may know these. They're really cool because they change color when you have a different pH, just like in litmus paper. And we put some of this in the water to see if it was excluded. They're a mixture of dyes, molecular weight approximately 100 or so, typically. And here's what we saw. So at the bottom you see a piece of naphthalene. And the major point of this slide is that the exclusion zone is right here next to Matheon, and all the beautiful colors that you see here, you don't see here. This is clear. It's clear because there's no dye there. The dye is excluded from the exclusion zone. So it looks as though, in terms of molecular weight 100 or so, that's excluded as well. We've also tried salts, and we have at least indirect evidence that the salts are excluded as well. So it looks like this is pretty general. I should point out, incidentally, but actually this is the most important part of the slide, that the color chart. So if you look at the colors that you see here, this color means pH 3 or less, and this is higher pH and so on. So it means that this region right here is just filled with protons or hydronium ions. Very high concentration of protons here, and that will be important in a moment. Okay, so the answer to the first question about generality is that many hydrophilic surfaces generate exclusion zones, and many solutes are excluded, so it looks like a pretty general phenomenon. Okay, question 2, is this zone really different from ordinary bulk water? I alluded to the possibility that it's different, but I presented to you no evidence. So I want to list the evidence, and I'm not going to talk about each one in detail because much of it's published and it would take too long. I'll discuss two of those pieces of evidence. So the list of evidence, as I said, much of it is published. The first one is that we used NMR and looked, for those of you who know it, about the T2 relaxation time. We found that the exclusion zone water molecules are more constrained than bulk water molecules. We used infrared radiation coming from the sample itself, and we found that the exclusion zone molecules radiate less. It appears their emissivity is lower, and therefore they're more stable than bulk water molecules. We found that this zone has negative charge, and bulk water doesn't, and I'll show you the experiments for that. We also found that a peculiar absorption in the ultraviolet at 270 nanometers using UV-Vis absorption spectrometry, and I'll show you that one too. We found it's more viscous than bulk water. We found that the molecules are aligned using measuring birefringence using polarizing microscopy. And we have some data of IR absorption, although not spectra yet, because you need a very thin sample showing that the molecular structure is somehow different. But we don't know from these results how it's different. And there should be another one. Nikolai Bumkin in Moscow is preparing a manuscript showing that the refractive index of this zone is about 10% higher than that of bulk water. It looks more dense. So let me just go over two of these, which I think are particularly germane to what I want to say. The first is the UV absorption spectrum, showing that 270 nanometer absorption. So the light is passed through a slit and another slit here, and we have an ordinary cuvette that goes with the spectrometer. And a piece of Matheon is put here, lining one of the walls, and we fill this with water. And we set it up so we can position the cuvette in any of a number of positions in this or this direction. Okay, so we can measure the spectrum far from the nucleating surface near and in the exclusion zone, etc. The idea is the exclusion zone absorption different from the bulk water absorption? And here's the result. So this is absorbance and this is a wavelength. And this is the distance from the Matheon water interface. So if you're more than, say, 500 or 600 microns from the interface, you're out of the bulk, out of the exclusion zone. This is flat, and flat means it's just the same as bulk water. As you put the light closer and closer and right into the exclusion zone, you see that there's a strong 270 nanometer absorption. So the next one is, and that's different from bulk water. The next one is that this zone has negative charge, and the experiment is shown here. So this is the inside of the gel, this is the outside of the gel. In other words, this is the gel, polyacrylic acid gel, and this is water. And we want to measure the distribution of electrical potential. So we took two electrodes, and these are, both of them are very fine microelectrodes used very commonly in biology, and the tip comes to a point less than 0.1 micrometer. So it has very good spatial resolution. And so we put one electrode out here somewhere, far away from the interface, and the other was the probe electrode, and we moved the probe electrode closer and closer to the interface. So if the probe electrode is far from the interface, that is 300 or 400 micrometers, the potential difference between here and here is 0, and that's what you expect. Now, as you get closer and closer to the interface, you begin to pick up a negative electrical potential. And that region of negative potential corresponds roughly to the size of the exclusion zone. So it looks like the exclusion zone has negative charge. Okay, so that's using the polyacrylic acid gel. Now we dumped the gel, and instead we put a piece of naphthalene here, and we did the same experiment again. And the result is shown here. So the electrical potential begins to become negative farther from the interface, and it gets actually to be more negative. And naphthalene has an exclusion zone that's bigger than the polyacrylic acid gel. So there's a good correspondence between the region of negativity and the region of the exclusion zone. So it looks from these measurements as though the exclusion zone has negative charge. Okay, so now if you think about it, you can say, well, this is crazy. It doesn't make any sense at all, because what you did was you took a gel, or a piece of naphthalene, you poured water, and the water is neutral. So how can you start with neutral water and wind up with this giant region of negativity? It doesn't make sense because the charge can't come from nowhere. However, another possibility is that somehow the water molecule is split up, and there's a region of negativity here, and a region of positivity somewhere out here. So the question is there such a region? Well, you've actually seen the evidence already for it. The evidence is shown here. You've seen this slide. I've turned it 90 degrees to make it correspond to the previous slide. So here's a piece of naphthalene. Here's the exclusion zone. And I've shown you from the previous slide that this region is a region of negativity. And remember, the pH value here is 3 or less. That means a huge concentration of positive charge. So you have negativity and you have positivity here. You have a splitting of charge. And if that's really true, then you expect to get current. That is, if you place one electrode here and one electrode here, connect them through a resistor, you ought to be able to get current flow. And here's a record of the current flow. So here's current versus time. And you can see that it starts at some high value, high in quotes, microamps, and goes down to some plateau value. It's not 0, it's a plateau value that's maintained for quite a long period of time. So that confirms that there is some kind of charge separation that occurs there. So basically, to summarize where we've been so far, we have some material here, and here is the interface between the material and the water. And this region of the water has the negative charge, and it has all these interesting properties. And this region here is a region of positive charge, where all those protons are. And so you have negative and positive, which basically means you have a charged battery in water. Now the idea of a charged battery in water is something weird that you really never think of. How is it possible that water can sustain a potential difference of this magnitude? Well, there's a good example of that that's shown here. And the potential difference between two regions of water here is immense. So there is precedent. So I've shown you in some detail these two, which I think are important. And if you look at the ones in blue, they tell you something about the likely structure of this zone. So the molecules are aligned, they're relatively stable and relatively constrained. If you add up those three, what you get is something that's like a liquid crystal. So at least that's the best sort of definition that we can think of. I'll tell you more in a moment. But so far, where have we been? We have something that looks like a liquid crystal. Not sure. It has negative charge. It excludes solutes rather profoundly. It may be non-dipolar, that's what I was alluding to, and I'll get to that in a moment. And one of the more important things is that it may extend pretty far from the interface, from the surface. And by far, I mean, we're not talking about 2 or 3 molecular layers, we're talking about 2 or 3 million molecular layers. Very, very far. And it was suggested 100 years ago by Sir William Harvey, he must have been important because he has Sir before his name, that water really doesn't have 3 phases as we all think of, the solid, liquid, vapor. Actually, there's a 4th phase. And there's actually a lot of old evidence for this. And I'm not sure if what we presented really constitutes a phase or not, but it's certainly different from bulk water. It's not a solid, it's not a liquid, and it's very extensive. So perhaps, now what about the real structure of this? I alluded to the possibility that just a stack of dipoles. And one of the reasons is that it's commonly thought that the ordering of water is simply the stacking of dipoles. And I thought so until 2 or 3 years ago. However, some very simple considerations led to a different kind of idea. The first is that, well, you know, it doesn't take a rocket scientist to figure out that this has negative charge and dipoles are neutral, so dipoles don't seem to work. The second is that this absorption at 270 nanometers, usually this kind of absorption is characteristic of ring-like structures, or at least structures with delocalized electrons, not dipoles. So dipoles don't really fit, something else. Another feature is that this is amenable, we found that this exclusion zone is amenable to shear thinning. And what do I mean by that? You have a surface, you have the exclusion zone, and you have flow that's parallel to the surface of this. And what happens is if you impose that flow, the width of the exclusion zone is narrower and narrower. It'll go down to 0.4 times the static size. So it looks like thin, and usually when something thins, it's a sign of a layer-like structure where you can erode layer by layer. So it's a suggestion, it's not really strong evidence. And the final point is that whatever structure one comes up with, you'd expect that it ought to have precedent. It ought to be something familiar rather than something weird and exotic that you've never heard of. So those are the kind of considerations that led us to think. And we thought, well, what about ice? Okay, is this maybe, we know the structure of ice, and is there some similarity of the structure of the exclusion zone to the structure of ice? Okay, so here's the structure of ice, and those red blobs that you see are oxygens. They're also hydrogens, and they're omitted for clarity. So in between these two oxygens, there would be a hydrogen here and a hydrogen here, and so on. So you can see, if you look at it from a particular angle, you can see that it's a layered structure built in hexagons. Now, if you look at the same structure from a different angle, from here, you can see that you can't quite make out hexagons as well, but you can see another feature that these are connected by these blue dots. Blue dots are protons. So it's very simple and elegant that the reason ice is solid is that these negatively charged oxygens are linked by protons. So you have minus, plus, minus, and that gives you something solid, hard, like ice. So this is the structure of ice. This is well-known structure, nothing controversial. The question is it possible that somehow the exclusion zone structure bears some similarity to that of ice? Well, ice doesn't work because ice is solid and this structure is not solid. But suppose you were to remove the protons from here, right, the crosslinkers. So you get two wonderful features. The first is that if you pluck away positive charge from neutral, it leaves you with something negative, which is what you want. Okay? So that's the idea. You remove the protons, you no longer have anything solid. You have something where the planes are not glued to each other anymore. The only problem with this very nice idea is that it's wrong. It's wrong because it flies apart. Because you see, you have an oxygen here and an oxygen here, and you take away this nice glue that glues these together, and then you have the negative repelling the negative, and that happens all over, and so the structure just flies apart, so it doesn't work. However, you can rescue this structure in a nice way. All you need to do, I've shown two planes here, all you need to do is to move one plane just a bit to the right. And if you do that, then the structures are glued together weakly. So, and for example, take this point right here. So you can see the oxygen back here, and in the plane that's nearest you, you see the hydrogen. So you have plus and minus, and they stick together electrostatically. So this can stick together. It can hold up. It's a weak connection, not a strong connection, which is really what you want. So this is a provisionally reasonable structure. And you can add up and compute the charge of this thing. It turns out to be -1. So it works in the sense that you get negative charge, you have something that sticks together, you have something that acts sort of like a gel that can be eroded. So what it would look like is nucleated here, and layer by layer this builds up. And you can see what it would look like if you were standing out here. something like that. So it builds layer by layer and such. Now, I built this thing by taking the upper plane and shifting it to the right by half the spacing between here and here. And now we have this glue that exists in many places. Weak glue, not strong glue. But I didn't have to shift it to the right at 0 degrees. I could have shifted it 60 degrees. In other words, in this direction or this direction. Any of 6 different shifts would have given the same result. So why is that nice? Well, that gives you a lot of flexibility. So what you can do is create different arrangements. So here's layer 0. Layer 1 is shifted by 0 degrees, 60 degrees, 120 degrees, and so on. And from that you get a helix. So why is a helix good? Well, helix is good because in biology, almost everything is helical. DNA, RNA, proteins, they all have helical structures. And they all need to interface with the water. And it's known that the water with which these molecules interface is ordered water. So it looks like this works. So the advantages of this non-dipolar exclusion zone model is it has precedent, that's ice. It has negative charge, which we need. It has ring-like structures, which could more easily account for the 270 nanometer absorption. It's able to accommodate helical solids adjacent to it. It's amenable to shear thinning because it's a layered structure. And this is a maybe subtle but very important advantage, is that large sheets eliminate Brownian instability. What do I mean by that? Remember I showed you in the second slide, I think it was, that when the dipoles line up, they're expected to be disordered when you get more than a few molecules away. And that is because if you look at the left side, each one of these is, let's say, a dipole, and each one undergoes some thermal motion. And by the time you get here, the thermal motion builds up so much that this brick-like structure will collapse. At least many people think that, others don't. However, this structure doesn't suffer from that disadvantage because we're talking about infinite sheets that extend many molecules in this direction and the direction perpendicular. And they're all glued together. This sheet is glued to this and so on. So you have a stable structure, almost like a brick wall. And it's not going to fly apart because of thermal or Brownian motion. So the large sheets eliminate the specter of Brownian instability. The only problem with the structure that I've come up with is that of polywater. How many of you have heard of polywater? Okay, so you know, polywater was you might say, a scandal that occurred, although that's not exactly the right word, that occurred 40 years ago, where some Russians, a guy named Deryagin, who was the most famous physical chemist in all of Russia, found that when we put water in these tiny capillary tubes, that the properties of the water were no longer like any water properties anybody ever heard of. They're more dense, hard to vaporize, hard to freeze, etc., etc. And here's a picture of Deryagin, and the structure that was arrived at is this structure. This is the same structure that we suggested. Now, this was arrived at by very different reasoning, using from Raman spectra and other properties, different reasoning from our reasoning. But this is the same structure, and therefore, I should mention that this structure appeared in a famous paper in Science in 1969 by Lippincott et al., who were chemists at the University of Maryland, right here. And this was a lead article in Science, and about 3 or 4 months later, some people discovered in the US and England and Russia that the water that was being used was actually not pure water, as the Russians had claimed, but actually had traces of silica leached from the containers, from the vessels. So it wasn't pure water. And then some Australians found that they took water and they sweat in the water, and they put salt in the water. They put salt in the water, and they found that the Raman spectra were very similar to what the Russians found. And so they concluded that the Russians must have been sweating in the summertime in their water, and that's why they got this stuff. So it turned out to be a big joke, and everybody laughed. And still, when you mention polywater, which the term, because it behaved like a polymer of water, people have a great big laugh. So this is the downside of what we found. That is, 40 years ago, it was suggested, based on the Russian experiments, which were not pure water. After all, what water is pure? Water is the best solvent we know, and it's impossible to get pure water. And this was turned down on that basis. And I guess I have to stop. my friend once had this pointer that got hot when you pressed too much, and so you had to stop talking. Okay. So it's physically distinct from the bulk. Yes, it's physically distinct. It's a liquid crystal and probably a layered honeycomb structure. It's interesting when you think about layered honeycomb structure, that the structure of graphene, for which a Nobel Prize was given last year, looks just like the structure we're talking about. Okay, now, can crystalline water explain various counterintuitive anomalies that we know about? Now, I'm not going to talk about the standard ones, I'm going to talk about some non-standard ones. Now, what behavior do we expect from a crystal? Okay, so crystalline elements obviously stick together. If you have a salt crystal, it's sticking together. And if you think about Jell-O, for example, Jell-O is 95% water. And I'm not sure if you ever thought about what holds the water in the Jell-O. So why doesn't the water just dribble out like a shower? And you know, there are some osmotic arguments to it, but there's another way of thinking about it, and that is if you look at the gel, think about the gel structure. It's either a polymer or a protein network, and in that network there are great big holes, and so what's in that network is really not pure water. It's what's inside there. These crevices are filled with the kind of liquid crystal I'm talking about. So this is something that is nucleated by these charged surfaces, and the layers build and build, and you have an exclusion zone water. You have exclusion zone water here, which is a liquid crystal. You don't have bulk water, and that's why it sticks. And if you think about why your Jell-O is like a gel, and most people studying polymer networks think about the viscoelastic properties of those networks and so on. But the water is gel-like. This kind of property that we've been talking about, this liquid crystal property, should act very much like a gel. So the reason that your Jell-O feels like a gel is probably because of the water. And I've handled some gels that are 99.95% water, just the barest trace of polymer, and it feels very much like Jell-O. So it's really hard to profess that the properties of this gel come from this trace network that holds it all together. Okay, crystals can be pretty stiff. Those of you who have gems of various sorts that you wear on your fingers and such. And so I come to what someone mentioned earlier, to this so-called water bridge. You see two beakers of water with a bridge of water in between, and the bridge is stiff because it hardly groups at all. And so when you think about how you can explain this, well, let me show you the video. It's more dramatic. You put two electrodes in, and you get this bridge of water, 10,000 volts, and you move one beaker away, and what happens is that the bridge is sustained, and it can be sustained for 3 or 4 centimeters, and it can be sustained for 30 minutes without any problem at all. It doesn't droop, or it doesn't droop very much. And this is not what you would expect from water. However, liquid crystalline water that becomes fairly stiff could conceivably explain this striking phenomenon. Okay, we found that crystalline water grows at the air-water interface, and so here's the air-water interface. This is taken from the web, and this coin is an old Hungarian coin, and just like a paper clip, it floats. Now, a paper clip, as you know, if you put it gently on the surface, it will float, but if you stick it underneath, it plummets to the bottom very quickly. So there's something different about the surface, and you'll say, well, I know what's different. The surface tension of water is higher, and that's true. However, you have to ask yourself whether the current explanation for the surface tension, which is that the single molecular layer of water at the top has more hydrogen bonds than layers below, whether that's enough to explain how you can support these heavy weights. So we began looking, and this is a small chamber, and it's filled with microspheres of water. That's why it's cloudy here. So here's air, here's a meniscus, and here's a clear zone. And this zone becomes clear after 15 to 30 minutes and stays that way. In about 24 hours, these microspheres sediment to the bottom. But this is a stable region here for many hours before that happens. So it's a clear zone. It has no microspheres, and clear zone is basically an exclusion zone. We measured, and we found it has a negative potential. And in the next slide, I'm going to show you in the video that this region here acts like a thick rubber band going from left to right. It's a cohesive zone. And I'll show you that here. It's the same picture as you've just seen, except we're going to take a probe and touch the surface and move it back and forth, and you'll find that the distance from here to here doesn't really change very much, at least noticeably, despite this mechanical perturbation. So you can see you're moving it back and forth, and that region, the dark region, is cohesive. It sticks together. So now if you think about what's holding this, this coin at the top, we're talking about many structured layers. We're talking about possibly a half a millimeter or a millimeter of this that's different from bulk water. Perhaps that's the reason why you can see that. It also explains, some of you maybe have seen this creature. This is a creature from Central America. It's a lizard, and that lizard lives near ponds, and basically the lizard walks on water. Therefore it's called the Jesus Christ lizard. And you can see how it scampers across the surface of the water. And the question you have to ask yourself whether this can be explained by one molecular layer or you need some thick region that's much more supportive. Okay, and also the same thing occurs with pendant droplet. So here's a situation in the laboratory where you have a droplet of water hanging from a pipette. It's about to be dropped, and it's about to be dropped on water. And remember, I told you that the water surface has this thick layer on it, but actually the same thing should occur here too, because you have water and air, so you have a water-air interface, same as down here. So I'm going to drop the droplet, and of course you know that when you drop a droplet of water on water, the two coalesce instantly. However, when you do the experiment correctly, appropriately, they don't coalesce instantly. They coalesce in steps. It looks a bit like a salsa. See, and that's been reported 100 years ago, so it's nothing new. We just published a paper, we thought it was cool, and we found some interesting stuff. And the reason is because the surface is different from the rest. You can actually see these droplets if you look carefully. So outside our laboratory, these are bicycle lockers. If you don't want your bike stolen, you put it inside, and it rains a lot in Seattle. So you can see there's water that's on here, and the droplets of rain, you can actually see them, that they sustain themselves. And if you look carefully, you can see this every day when it's raining or stuff. So they don't coalesce instantly. The reason is because of this EZ that's around them. Another feature of this exclusion zone is that it keeps out the protons. So this is, if you think about this is very strange because you have negative region here and positive region. And so whenever you have negative and positive, what's going to happen is the two will combine and annihilate one another. This doesn't happen because you can measure the electrical potential difference over a long period of time and it's sustained. So why doesn't it happen? Well, it doesn't happen because this exclusion zone here is made up of a very dense network. And this dense network is so dense that, of course, it excludes almost everything. It also excludes the hydronium ions, which are really positively charged water. These holes are too small to let them penetrate, and therefore, the separation, positive charge here is maintained. So the battery charges remain separated. So the answer to question 3 is yes. Liquid crystalline water explains many anomalies and explains why the water battery charges remain separated. Okay, so what charges the battery? It took us an embarrassingly long time to figure out what was going on. It turns out that it's the sun. It's incident radiant energy that does it. And the first observation we made of it is to take a piece of Naphion, the exclusion zone, the microspheres, and someone took a lamp, one of these gooseneck lamps, and shined it on the chamber, and we saw that the exclusion zone grew bigger. By the way, the green color is just because we use green light in the microscope. It has no other significance. So you can see how it grew here. So we decided to do some experiments, and we started with the control. Here's an exclusion zone, here's microspheres. And then we took an additional light using an LED, light-emitting diode. This is about 1 micrometer wavelength. And we shined it on there for 5 minutes, and you can see the exclusion zone grew by between 2 and 3 times. Very powerful effect for a very weak illumination signal. Someone said, oh, this is a mistake because you're using infrared, so the chamber heats up, and you're just measuring the secondary effect of heating. And so we took a temperature probe and measured at the end of 5 minutes the temperature, in different regions, and we never saw more than 1 degree. This is very weak. So it's a direct effect of the photons that create the energy. So, of course, we wanted spectral information, and here's the exclusion zone size versus wavelength. This is the UV region, the visible region, and the infrared region. So, for example, we'd set up an exclusion zone, turn on a green light, and find that after 5 minutes, the exclusion zone would increase by 60% or so. So what the result shows is that it depends on wavelength, and the expansion, the light expansion depends on wavelength, and the infrared is particularly important, especially at 3,000 approximately nanometers, or 3 micrometers. And it's well known that 3 micrometers is exactly the wavelength that's most absorbed by water. So the wavelength that's most absorbed by the water is the wavelength that expands the exclusion zone the most. This underestimates the real effect. The reason it does is that the LEDs that we had available in the infrared region were very weak. That is 1/600th of the intensity that we used in this region, low intensity. If we had used the same intensity illumination for these experiments, then this whole curve would be way up above the roof. So if we had, the way we should have done it, to use the same intensity throughout, then probably this curve would continue, and would go way up at 3 micrometers. So what does that mean? It means if we did the experiment here, turned off the visible light, it would make almost no difference. It's the infrared that counts. That counts more than anything. And the infrared is all over. It's coming from the walls, it's coming from the floor, it's coming from me, from you, from everything. It's almost impossible to get rid of it unless you go to absolute zero. So the conclusion is that incident radiant energy is the energy that builds the exclusion zone and separates the charge. It's free energy, literally free energy that comes as a gift from the universe. So the answer to the question about the energy is that the easy buildup is powered by photonic energy. It orders the water and it charges the water. So now if you think about an energy flow, I guess you might say Earth instead of universe, although the same may apply. The standard picture looks like this. So the sun gives its energy to the water, and we can go swimming in the summer because it heats the water. Okay, so we all know that route. And what I've suggested to you is a second route, a distinct route, that is the sun, the light, the infrared, hits the water, and it imparts energy for building order, building that building order and separating charge. Okay? It's not clear which one is more important, whether this is more important, or this is more important, or even possibly, you have to go through this to get to this. That's another possibility. So, you think that, okay, he's told me that the sun, or energy, is hitting the water and giving energy to the water. Now, if this energy starts building up, what happens to all this energy? The water doesn't explode, right? Something has to happen to all this energy. Where does it go? And we found that this energy can be harvested, actually, in various ways, some of them surprising. So this was a surprise for us. We took a container of water. Just a student came running into my lab, to my office to tell me this. So we took water. We put a tube in here. At first, the tube was made of naphthalene. That is that polymeric material that gives very nice exclusion zones. We put it in here, and the student had put some microspheres in here to just see what was going on. We put a microscope lens down here to look inside the tube, and what he found was persistent flow through the tube. Incessant flow through the tube. Now, this is impossible because there's no pressure gradient driving this through the tube, right? But he found it consistently. So here's an example. This is with the naphthalene tube. You can see this is flowing through naphthalene, exclusion zone, naphthalene, thin tube, about 600, 700 micrometers. And we found the same thing with a gel. So instead of the tube, we took a gel, polyacrylic acid gel, which also builds exclusion zones, made a tunnel through it, and filled it with water and some microspheres. And here's the gel, here's the exclusion zone, and here's the microsphere flowing through. We even have some preliminary data showing that if the water is higher here with a dam, still it continues to flow. We don't know each day whether it's going to go from left to right or right to left, and we're just studying this, but it flows. So what does this mean? Well, unless you have a perpetual motion machine, it's evidence that somehow you have to have energy, and that evidence that incident energy that I told you about, it must be absorbed. There must be some energy that comes from outside, and it can be harvested to do work. Okay, so that's nice. But again, you really don't think about water as absorbing energy. If you have a glass of water here, you think of it as being in equilibrium with the environment, and I'm suggesting it's way out of equilibrium, and it constantly absorbs energy from outside and transduces that energy into what could be useful work. So, but if you think about it, you have a glass of water, and I'm suggesting that that's happening, and you know, this is a shocking kind of concept, but if you think about it, think about the plant, which is mostly water. What does the plant do? It absorbs energy from the sun and transduces that energy into useful work, that is, metabolic work, bending, growing, whatever. And I'm suggesting to you that the same thing happens with the water as happened with the plant. It's no surprise because the plant is made of mostly water. So, question 5, why is all this important? I think it's foundational, it could be foundational for any kind of science involving water and light, and that would tie together chemistry, physics, biology, etc. So what I've really shown you is kind of summarized in a nutshell here. You have a charged particle or molecule sitting there, and I've shown you that surrounding it is a rather large mass of liquid crystalline water, negatively charged, and surrounding that are all the positive charges that are running around in the water. So this is the real situation. The textbook says that what you really have is just this, and maybe 1 or 2 neutral water layers. And this is so very different, what I've shown you, if it's true, so very different from what the textbook says that you really need to consider, reconsider many of the results that occur in aqueous chemical reactions, not only in the laboratory, but also in the cell, because this stuff, this is my 2001 book, and this kind of water just fills your cells. There's much evidence for this, and I don't want to take the time to go over it, but what I've told you today applies throughout your cells. Now, so here's a question for you to tickle your intellect. Suppose you have 2 particles instead of 1 particle, okay, and you have a glass of water, and both particles have the same charge. And you take the 2 particles and you drop them in the water, and they're pretty close to one another. So they both have the same charge. What happens to the distance between them? Anybody venture a guess? Separate. Everybody agree? Okay. What happens is they come together. And thank you. Like likes like. Right. This came from Richard Feynman. I think it came from his Nobel lecture. He said the reason for this paradox is that, first it's like likes like, because these are like charges, and they like each other, so they come closer together. And the reason that happens, he conjectured, is that there's an intermediate of unlike charges, so like likes like because of an intermediate of unlike. And that's what happens when they start coming together, because this provides an attractive force that pulls the negativity and pulls the negativity, and it stops. And they do this, and it stops when the attractive force equals the repulsive force, that is, the force negative and negative repel each other. So then it stops, and the 2 are stable with some separation. And if you have many of them, then you get a situation that looks like this, which is known as a colloid crystal. And probably your yogurt is something like this, with particles separated from each other for the reasons I was talking about. Okay, so now we've got to come back to the cloud, and we ask, why does the water vapor coalesce into discrete units? Okay, and a possibility is that it's the like-like-like mechanism. The aerosol droplets that are throughout the atmosphere have the same charge, so you'd expect, of course, that they're going to repel each other and disperse all over. But you know that doesn't happen. They actually come together, and they come together, and the energy for bringing them together, despite their like charge, is the sun. The sun is out there doing it. So I think this is a possibility to explain why on a nice summer day you can see puffy, discrete clouds separated from one another. A special example of the like-likes like, I think, is the origin of life. And you ask the question, well, how did it all begin? What's step 1 of the origin of life? And I think it's that the Earth may have been filled with molecules of some sort all over the place, but you can't have a cell or anything unless they come together, right? And so you have to have coalescence. So you start with something that may look like this, some molecules in water, you add sunlight, and what happens is they come together because of the light mechanism, just like the cloud. And the aggregate keeps growing just like the cell. It's a kind of self-assembly. It has negative charge just like the cell. It has potential energy just like the cell. And it has even the capacity to divide. If you change the local pH here, they'll come apart. So step one in the origin of life might be a simple light-induced aggregation. All you need for that is the sun's energy and molecules and water. And if it's as simple as that, if step one is as simple as that, then the question arises, is life being created continually? And in step one, it did not happen only one time 2.7 billion years ago, or whatever it's supposed to be, but is it happening all the time? Step one in the origin of life, even today, outside. Now, another example of this is that just came to light recently is energy from food. Okay, so this seems to be a pretty obvious kind of thing we all know, we all understand. So for lunchtime, we might enjoy something like this. I understand there's a McDonald's right nearby. And so the question is, well, gee, this gives us energy and builds us. And where does the food's energy come from? So those of you who know biochemistry will know that, oh, this is very simple. It's the protein and the fat, whatever, gets metabolized, gives us energy, and makes us fat, right? However, if you go inside the hamburger and figure what's inside this hamburger, well, here's protein or sugar, fat, whatever, and surrounding each of these is an exclusion zone with negative charge. And surrounding that are these positively charged entities. So it's certainly true that if you eat too many hamburgers, that the protein here adds mass to our bodies and we get unproductively heavy. However, it's not necessarily the same as where we get our energy from. The question is this. Since we have charge separation, negative and positive throughout here, and this charge separation represents potential energy, the question is, and you know that if you take in potential energy, it can't vanish, it has to do something. And the question is, how much of the energy that we get from the hamburger comes from the charge separation in the water? What I told you. So, how much comes from the water? And the same thing is true, of course, here and the juices and whatever. How much of the energy comes actually from the charge separation in the water? And the same thing here. If there really is exclusion zones, are exclusion zones here, they contain potential energy, charge separation. The potential energy doesn't vanish, it does something. And the question, which is not answered, it's just a speculative issue, is how much of the energy do we get from the water? Sometimes we're tired, hot, we drink a nice cool glass of water, and suddenly we're filled with energy again. It's actually the same as walking in the sun. You know, the sun, I didn't mention that before, but of course, the sun hits us, we're full of water, the sun builds EZ water, and we go out in the sun and we feel energized. Is that really purely psychological? Or does the energy come because the sun is being absorbed, builds the EZs and separates the charge, gives us energy? Okay, one final implication, and then I'll quit, is an EZ phenomenon. Again, something that's come up fairly recently, and I think appropriate for this group is about electromagnetic signals containing information. So we have a situation that looks like this, and this is a very regular structure, and therefore it would look as though it has no particular information to convey. Now, the nucleators, if you look at the nucleating surface, typically, this is shown here, a sort of generic one with positive and negative charges displayed on the surface, and there's no real information here because it's a very, very uniform surface. However, this doesn't really exist. Every molecule has a particular charge distribution, and it's not like this. It's not as uniform. Typically, here's an arbitrary example where you're missing a few negative charges. So this is substance A, okay, missing some charges, and it's going to nucleate this EZ structure. And the EZ structure also will be correspondingly missing some of these oxygens as a result. So this structure then contains information from the nucleating surface. And this information can project over many easy layers. So you have this building of layers, and each layer is missing a few of these that correspond to some missing surface charges. Okay. So then, inside the water, if you have these exclusion zones that persist, and we have evidence for long-term persistence, then you have information that's separate from the nucleating surface and actually stored in the water. And so if you think about it, you have electromagnetic energy that's being absorbed by the water all the time. And I suggested that this water contains these kinds of structures with information in them about the nucleating surface that gave rise to this particular structure. So this structure is vibrating because of all of the electromagnetic energy that came in. And the vibrations here are going to be different from the vibrations everywhere else. And so basically, the output from the water contains information. Therefore, the water may contain information. And I don't know if some of you know about the recent results of Luc Montagnier, Nobel Prize winner. He's studying water now, and he comes, by the way, to our water meetings now each year in Vermont in October. Some of you may be interested, very interesting meetings. What he found is he put DNA in water. Okay, and it's a closed flask. And next to it, he put water. He put them together for 24 hours, and he suggested that the DNA is informing the water. Okay, then he takes the water that's been informed, and he uses this to make new DNA, using, some of you know, the PCR reaction. And the new DNA that's made has the same sequence as the original DNA. Okay, and so he concluded that there's some electromagnetic signal that's transmitted from one flask to another. This is not as new and radical as you might think. In Russia, Gurevich in the 1930s showed the same. He showed something similar with onion root cells that were undergoing mitosis. So he had two preparations widely separated from one another, and they'd be undergoing mitosis at their own rate. If you bring the two flasks close enough together, they would synchronize with one another, like women's menstrual cycles. So they're synchronized, and then if you pull them apart, they're not synchronized anymore. And if you kept them together and you put a lead shield, they're not synchronized. And you remove the shield, they are synchronized. So this is actually practically 100 years old, with many studies that have shown electromagnetic signals can contain information, important information. This is the frontier. So the water then has the capacity for memory and the capacity for electromagnetic transmission of information. So I've been here. We learned a lot about water over the past decade or so, and lots of people doing these studies, ranging from visiting faculty members to postdocs, students, many undergraduates who really like this stuff, and everybody is trying to understand about this molecule, and they're all deeply immersed, so to speak, in the water, and so is the... Anyway, thank you for being so attentive. Yeah, the question is whether EZ can be found in water, in just a glass of water, and the answer is yes. And see, it's very easy to measure this. We can measure it because exclusion zone absorbs at 270 nanometers. So you take a cuvette of water, and if it mostly it shows no 270 nanometer absorption. However, in certain cases there is a 270 nanometer absorption. For example, healing waters from Lourdes, from the Ganges, have very strong 270 nanometer absorption. And we think that the reason they're healing waters, and the reason they actually work, is they contain some of this special kind of water that's in the water itself. Even the glass can nucleate this kind of structuring itself. So there are many possible sources to give rise to this, and these can persist over many months.
Could this explain homeopathy then?
Well, yeah, that's what I was getting at. See, homeopathy is, is a very controversial issue. I don't know how many of you know about the field of homeopathy. You take a substance and you dilute it, say 1 to 10, and you shake it. It dilute it 1 to 10 again and shake it, and keep doing that 100 times. And by the time you've done it 20 or 25 times, you have no original molecules left, only water. However, and so for most people, homeopathy is obviously a stupid artifact that couldn't possibly exist. However, it's been demonstrated, first of all, in terms of medical efficacy, that it really works, and it works on animals too, and it works on culture. So it seems that it's not an artifact. How it works is another story, but it certainly involves, if it does work, it involves this kind of, I think, easy structure that exists. In fact, some people, I mentioned Montagnier's experiment, he didn't really use DNA in water. He used dilutions of DNA in water because he got a stronger signal when he used the dilutions. And the reason he got a stronger signal, he thinks, is because this special kind of structured water is building up each time you do the dilution. And because you have a lot of this easy water, you get a stronger signal. So, yes, it's certainly related to homeopathy.
A couple questions about the experimental sensitivity of the mechanism. What happens if you change the charge on the boundary? And what happens if you use...
Let me answer that first. Okay, change the charge on the boundary. We tried that. If you put a negative charge on the boundary, you get a bigger exclusion zone. If you put a positive charge, you get a smaller exclusion zone.
How about changing the dipolarity of the liquid?
At first we thought that this is a phenomenon that's specific to water alone, because obviously water and biology and such. Pardon? No, but we were shocked to find that it was not. You were not shocked, but we were shocked because we thought there was something really special about water. We tried ethanol, methanol, DMSO, and a few others. We saw this kind of zone. It was not as big, and we found it also next to certain metals, to our surprise. So it's a kind of general phenomenon, which scares me a little bit because in thinking of this structure of water, I presume that there is something similar that occurs with these other molecules.
And maybe most importantly, how about the pH of the liquid?
If the pH of the liquid, you see, I hesitate to answer the questions of pH because this creates its own pH, right? In other words, you start with neutral water, and the pH of the material just beyond the exclusion zone is very, very low. So we start with almost neutral pH, it's not really neutral. If you start with water that has, for example, with HCl in it, to reduce the pH, we find that if you reduce the pH enough, you create a lot of protons, that it inhibits the growth of this structure. And you use the opposite, you use sodium hydroxide and it gets bigger. So yes, there's definitely an effect of pH.
And you showed a graph of the effect of absorption of different wavelengths on the size of the exclusion zone. As far as I remember your graph, the minimum effect on the width of the exclusion zone was in the ultraviolet, and yet you previously said that was the strongest absorption. Could you explain why that happened?
No, I can't because we're confused. I mean, that's a good question. This absorbs at 270 nanometers. And we'd expect that something has to happen because there's energy that's absorbed. We're trying to make the measurements now, they're not made yet. I think that this has a particularly strong effect on the charge separation rather than on the size of the zone. Well, we don't know that for sure, but it's a very good question.
One of the most interesting properties of solutions is the solutions of is the ability of solutions to rotate polarized light. Those chiral solutions, which goes back to an experimental history going back a while. And what's so interesting is when you think of crystal structure in a macroscopic scale, say rotating polarized light, it seems a little more intuitive. But when you dissolve that in a solution, and you've got supposedly dispersed solutes, and they're still capable of organizing that incoming electromagnetic radiation in such a discrete way, it seems like a total paradox. I'm wondering how much thought you've given to maybe how this approach could shed some light on this.
I think you're onto something important. So a possibility is that if you have easy nucleating molecules scattered throughout the solution, you get order. It's possible that the order around one of these molecules, there's some coherence in the order among the different exclusion zones. In other words, you have one exclusion zone, you don't have one exclusion zone that's oriented this way, the next one this way, the next one this way. Somehow they all come into coherence over a very large domain. That's a possibility I think, to explain what we're talking about. And the very nice evidence for that comes from Mei-Wan Ho, where she runs the Institute for Science and Society. And her book, a really nice book called The Rainbow and the Worm, has pictures of worms. And she has a very sensitive measure of birefringence. And she shows that macroscopic regions of a worm actually have the same degree of birefringence, or the same orientation, macroscopic. So you have one region of blob that's oriented this way, the next blob of the worm is oriented this way, and so on. So she shows evidence of very large domains inside biological specimen where the orientation is the same direction. I think that maybe get sort of an answer to your question.
I'm thinking about structured water that's around cancer cells. And you have an opportunity, is there any chance that the EZ is similar to the structured water that we're talking about that's around cancer cells?
There's been a lot on this, and I haven't followed all of it, but it's certainly possible. Many years ago, Gilbert Lane, who was a pioneer in this area, he's written 5 books on water in the cell, basically. It's where I got my start. This kind of a pioneer on whose shoulders everything I present is now in his early 90s, and still moderately active. And he showed that cancer cells have less structured water than non-cancerous cells. I'm not sure if I know about the water around the cells. Are you talking about the work of Mortenstein or something? Or
No, I have not been able to get it. I'm not that into it. I've just recently begun studying and looking into it. And I was just curious if this was similar to the EZ, whether by my structure or
well, I mean, it's possible, I think, from our evidence that there's only one structure, and that's EZ structure. We may be wrong, but we have so much evidence now about EZ structure. So when people talk about structured water, I think of EZ, and I think it's likely, although we haven't proved it, that all structured water is this kind of water. Where it is not so clear, but it's a new frontier for all of biology, because cells are filled with this stuff. Pathological cells, like cancer cells, probably have much less of it because the order is diminished.
Okay, I have a couple of basic questions. Number one, when you talk about the air interfacing acting like the polymer surfaces or the ones that you say, is that saying that air is a hydrophilic surface and or have you tried with other gases? And is there a similar amount of time to grow the exclusion zone with air as opposed to other materials that you work with?
Well, that's another good question. Everybody thinks about the air as being hydrophobic, not hydrophilic, and I suggested to you that this grows where things are hydrophilic, and so this would seem to be an exception. So there are either two possibilities, I guess. One is that the air is maybe hydrophilic, if you want to think about it. It's filled with vapor, you see, and vapor, water vapor, you think, well, that's hydrophilic. Or it may be that the generality that I suggested is not correct, and it's not necessarily just hydrophilic surfaces. Actually, I didn't have time to present the slides, but the air-water interface is actually rich with structure and information. It's not just a layer like that. You can actually, if you look down at warm water, for example, with an infrared camera, or even with your naked eye, if you're good at looking, you can see a mosaic structure on the water. This mosaic structure is rich with information, and it has something to do with how the water evaporates as well. So I just barely scratched the surface. I wanted to show slides, but I was given 1 hour, so I didn't want to overdo it.
And then the other question was, when you showed the students, looking at a tube under a polymer and the flow of basically water through the tube, is that related? And I guess I'm just wondering about capillary action and how big that tube was and whether there was plants are using this. It's not only capillary action that plants use to water.
Yeah, you're right on. This is exactly what I think is going on. The capillary action in plants, you know that it's not so clear how a redwood tree, 300 feet tall, how the water draws out, because there are emboli that are caught in between. Nobody really has the answer to it, but I think the answer has something to do with the charge separation and the exclusion zone. We know that the vessels in the plants, the xylem, actually have exclusion zones because we've seen evidence, actually from Martin Candy in Australia, that you can actually put particles in the xylem and you can see that they're excluded from the angular region just inside the wall. So there is an exclusion zone. If there is an exclusion zone, it's negatively charged, and that means the core of the vessel has positive charge. And if this positive charge, I think that the repulsion forces between the hydronium ions, it actually pushes upward. So I think that's the reason why you get capillary action. It has to do with this and trees.
In my mind, with the heavy water, would we get
Well, yes, we've tried E2O and we do get an exclusion. It's a bit smaller than you can see here, but we get it. The purest water that you can get gives you the largest exclusion zone. We found that almost anything that you would put into the water makes the exclusion zone smaller rather than larger. So it actually is not clear yet what determines the extent of the exclusion zone, because if you think about it, if you're building a million layers, then what's the difference between a million layers and 1,050,000 or 1,500,000 or something like that? Once you build so many layers, why shouldn't you get even more layers? And we're trying to figure out now what does limit the size. And a possibility is that protons or hydronium ions that accumulate just beyond the edge of the exclusion zone, that's the limiting factor. When you get too many of those, too many positive charges, you can't build out anymore. Not sure of that, but that's what we're studying right now.
I'm sure you've probably heard of the work of Rustin Roy, material scientist who wrote an interdisciplinary paper in 2005 on the structure of water, and he claims it's a customer process, pressure changes, that, there's no molecular change. Yes, there was a significant change from the nano-level scale. Have you noticed anything in that regard?
Yeah, I knew Rustin Roy very well, and of course, he'd come to our meetings on water many times. And his death is very sad because he was a real pioneer. And he thought that this sort of easy phenomenon was inevitable, because the same thing happens in semiconductor growth, for example. You have a template and epitaxial growth that grows up very longly. He said water must be the same as that. And so he was absolutely sure that this would happen. Regarding the succussion, so what the succussion does is it, I think we're studying this right now, shaking. What does shaking do? Well, shaking is interesting because you're then creating air-water interface all the time by mixing the water with the air and a bubble. Again, I don't have time to talk about it. I think a droplet and a bubble both have an easy shell around it. So by succussing, you're creating more easy material. At the nano level, even micro level, and maybe sometimes larger. Right, I think that's the case. And we found that when we shake, we get 270 nanometer absorption afterward. So it looks as though that really is happening. That shaking gives rise to more easy.
Your comments on biology are really interesting. So there's the whole issue of microbial colonies, algae at the surfaces, and membranes. And have you looked or heard much about toxicity, toxicology? Because in toxicology, control over colloids seems to be really important. Repeatability of testing.
Well, we haven't done much ourselves, but just last week I was with Eshel and Jacob, who's been doing many experiments on bacterial colonies, and they organize themselves sometimes in very nice arrays, and
Signaling and things like that.
and signaling. But one issue is that sometimes you can get rid of it, you can kill the bacteria, you can kill it by changing the pH. So if you have certain surfaces, and these surfaces generate exclusion zones, and they generate many protons beyond. Those protons can be lethal for the bacteria. So the killing that you can witness when you have the right kinds of surfaces could be a result of those huge numbers of protons that come from the exclusion zone. That's what I'm thinking, but we've not studied it.
Did you bring your book?
No, I mean the Sales Yale's book. You can buy it, it's cheap on Amazon, it's slightly more expensive on the book website.
All right, I thought of another question. Is it possible to, either with electric charge or some, I guess I'm not real clear on how you get protons out of water, but you've created this exclusion zone, you've moved a lot of the protons over to one side or the other of the water. Can you then apply potential or some way of pulling the protons out to even further? In other words, can you make like a full vessel of exclusion zone water?
Yeah, I mean, I think what you're alluding to is that these protons are mobile protons. They come, just to clarify, you start with water, you wind up with a negative zone and a positive zone, so somehow the water has split. The process of splitting is not so clear, but those protons are free. They repel each other. Because they repel each other, they want to disperse infinitely. And when you think of the different flows that occur inside your body and every one of your cells, the simplest mechanism to do that is to have these protons, because they repel each other, they want to flow away, and they're attracted toward negative surfaces. So you have every opportunity to flow here. Also in your blood vessels, if you think about it, your blood capillaries, the capillaries are sometimes smaller than the red blood cells. It's a lot of resistance to push those red blood cells through. And the question is, where does the energy come from to do it? We all think, well, it must be the heart. But people have calculated that the pressure that you get from ventricular contraction, you need something like 1 million times the pressure to drive those red blood cells through the capillaries, because the cells are bigger than the capillaries. It's like stuffing a soccer ball through a narrow pipe. It's very hard to do it. However, I pointed out to you that infrared is very powerful in building these exclusion zones and separating charge. So it might be that the infrared that comes from all the metabolic reactions that are occurring in your body, that this energy is recycled. It's not just radiated away. It's recycled into your cardiovascular system to help push the blood through the vessels. So I think there's a lot of flow phenomena that you were alluding to that occur as a result of this sort of thing.
Have you measured the size of the exclusion zone with different electromagnetic gradients on the liquid, oriented different ways and different strengths?
I'm not sure I understand the question. Electromagnetic gradients, maybe more specific than that? We've tried magnets.
Well, this seems to be a question of balancing charges, and if you create a gradient across the entire liquid, the question is, does that change the charge distribution, therefore change the size of the EZ?
Everything you do electrically has an effect on this. We even put magnets, permanent magnets, on it. But first of all, if you put an electric field, an DC electric field, of course you get many changes. Protons will move toward the negative charge, the exclusion zone will grow or shrink depending on but we found that permanent magnets have an effect as well. We can create regions, isolated EZ regions that have either a negative charge or can actually create positively charged regions. If you put a magnet right there, these regions move either toward or away, toward the magnet or away from the magnet. And we're now studying these effects because they're paradoxical. You may know regarding magnets that the work of Andre Geim, he's the fellow who just won the Nobel Prize for graphene. 10 years ago, he was lifting frogs in magnetic fields and lifting water in magnetic fields. So there's definitely an effect that occurs. And a Japanese fellow demonstrated that you can actually take a trough of water, put some red dye in, the red will become obvious as well. We put a superconducting magnet behind the trough of water and split the Red Sea so you can actually walk through. So magnetics, magnetic fields have every conceivable kind of effect on water. And, you know, the idea that we wear a magnet or wear a bracelet, and most people poo-poo any kind of effect. We see magnetic effects on water and exclusion zones on a daily basis, so something's going on. What it is not exactly clear, but it's clear that major effects are occurring. Well, what I meant is that you can have layer 1, layer 2, layer 3, and so on. The first layer is here. The second layer can be shifted this way slightly. The third layer can be shifted more, and then you get a regular, slightly angled structure. But it doesn't have to be that way. You can have the first layer could be shifted this way, the next one this way, the next one this way, the next one this way, and then you can build a helix. The helix could be a regular helix, it could be an irregular helix, it could be almost anything because you have many degrees of freedom. And so I was just suggesting that this is convenient if you have large molecules lined up right next to the water because you can accommodate many different kinds of structure. So does that answer your question? Or maybe not. I'm sorry, what is the structure that...
I haven't looked at what it is.
No, we haven't, but that's the frontier. That you mean what kind of layering you have? We presume that if you have a molecule in the water, that somehow the interaction between the molecule and the water will give you a certain result, and therefore there'll be some kind of imprint in the water of what that molecule was. And that imprint is information. That information is really important. Yeah, so she's referring to the work on freezing of water, and if you say words or sounds to the crystal during the freezing process, you get crystals of different shape. And if you say, I hate you, or play loud rock music, then the shapes are ugly. And if you say gentle words like I love you, then you get beautiful crystals. I mean, this is not really scientific work because you have to repeat this thing again and again and show the same sounds, the same crystal structures, and I don't believe he's done that. However, we have evidence that sound and ultrasound both affect the EZ, and if the EZ is somehow related to ice growth, which we think is the case, then it's conceivable that he's right on, that we really can get some structures using audible sounds. Well, right now we're working on sound. We take an exclusion zone, we play sound, we try ultrasound, and we measure the size. With ultrasound, 7.5 megahertz, the same as for body scans, in fetus, for example. What happens is you turn on the ultrasound, the exclusion zone first gets a little narrower. As soon as you turn it off, it grows 5 times its original size, and then diminishes. So we don't fully understand it, but it definitely has an effect. And sounds also, certain frequencies can expand the exclusion zone. And well, no, we haven't looked at it specifically. Maybe we should get